So the next mini seminar we have is from Zhaozhi Qian. Zhaozhi is a PhD student who joined our lab in 2019, and his presentation is on discovery using machine learning. Zhaozhi, if you're able, would you mind sharing your screen?
So hello everyone, I'm the presenter Zhaozhi, and I'm really excited to present to you our recent work titled Integrating Expert ODEs into Neural ODEs, Pharmacology and Disease Progression. So this is actually one of the first of a series of work that is devoted to making scientific, enabling and accelerating scientific discovery. For this particular work, the main motivation is to bridge the gap between the research lab and the clinical practice. Many physiological and pharmacological models have been developed by the scientists in the laboratory settings. In particular, we will consider the wide range of pharmacokinetic, pharmacodynamic models, or PKPD models. The PKPD describes the temporal evolution of certain physiological variables under the influence of medication. And here, as illustrated in this figure, the variable A represents the treatment, and the variable E represents the physiological variables. The temporal dynamics is usually specified through a system of ordinary differential equations developed by the modelers. In particular, the physiological variables are carefully chosen and medically meaningful. These variables may include, for instance, the drug concentration in the plasma or the concentration of cytokines in the immune system. However, the physiological variables are usually defined at a very low level, molecular or cellular level. And furthermore, they are measurable in the research labs, but not routinely observed in the clinical practice for various constraints on the measurement setting. In contrast, in the clinical setting, as illustrated below, the clinicians often base their decisions on clinical biomarkers and endpoints. These variables are defined at a much higher level, organ or body level. Examples include lung mechanics and survival. So these clinical endpoints are not often modeled by PKPD due to the high complexity of the underlying biological process. So in summary, the low-level physiological variables are observed in the research lab, but not in the clinic. Hence, they cannot be directly used to inform bedside clinical decision. On the other hand, the clinical biomarkers and endpoints are crucial for decision-making, but they are not modeled by PKPD. So with that background in mind, this project addresses two interrelated goals. And the first goal is to use machine learning to bridge the gap between the lab and clinic. And specifically, we would like to establish the link between the physiological variables and the clinical endpoints. And the second related goal is to use the scientific knowledge encoded in PKPD equations to inform machine learning systems to make principled predictions. The existing approach to model clinical time series and disease progression are often based on the so-called latent variable models, which is a pure machine learning approach. It introduces a set of latent variables, Z illustrated here, to capture the complex dynamics of clinical biomarkers and endpoints. The learned latent variables, however, do not correspond to any known entity or may not have any semantic meaning. Moreover, the method does not take into account any existing scientific knowledge, including the ones that we know for PKPD. As a result, this approach is often not sample efficient, which means it can be difficult to apply to rare or novel disease where the samples are limited. The hybrid modeling approach attempts to combine a given expert model, such as the PKPD equations, with machine learning models. However, our problem setting introduces challenges for conventional hybrid models. Essentially, these models attempt to use machine learning to correct a misspecified expert model. For instance, they can be helpful when the expert model assumes a linear relationship, whereas the true phenomenon is nonlinear. So it is based on the assumption that the expert and the machine learning model is predicting the same set of variables. And it further assumes that the prediction targets for both models are observable. However, neither of these two assumptions hold in our setting because the PKPD does not model clinical endpoints and the physiological variables are not observable in the clinic. Our proposed solution is the latent hybridization model. It's a novel framework of hybrid modeling. It combines pharmacology PKPD model and the machine learning latent variable-based model. The hybridization occurs in the latent space. It uses two sets of latent variables, the physiological variables that are driven by the ODE, the known ODEs given by PKPD models. It also includes a set of fully machine learned latent variables. Here we specify that these variables will be driven by a neural ODE. And both sets of variables will influence the clinical endpoints nonlinearly. In order to train and make inference about latent hybridization model, we use Bayesian variational inference. So we designed an encoder network to infer the variational distribution of latent variables and physiological variables from the historically observed measurements and outcomes. And then we use a decoder to extrapolate the latent path over the entire time horizon. And in this case, since our models are ODEs, the decoder are naturally just numeric ODE solvers. And finally, we learn a mapping from the latent space to the outcome space, to the clinical endpoints that we care about. And the entire system can be trained end-to-end using adjoint sensitivity gradients. In addition to extensive simulation studies, we performed a case study on dexamethasone treatment for COVID-19 patients in the ICU. Dexamethasone is one of the first drugs approved for COVID-19 treatments, and the PKPD of dexamethasone is well understood. The drug has an immunosuppression effect and can be used on patients with cytokine release syndrome. However, if the dosing of dexamethasone is too high, it may oversuppress the immune system and become detrimental to the recovery. So it is very important clinically to keep the level of immune system right. And here, the level of immune response is a physiological variables modeled by PKPD, but it is not observed in clinical setting. We apply latent hybridization model here to infer the immune response from observed biomarkers. We use a range of biomarkers, including general inflammation markers such as C-reactive protein. The inferred trajectories of immune response profile have been qualitatively validated by clinicians. And we also quantitatively show that the latent hybridization model achieves better prediction accuracy and sample efficiency than conventional latent variables or conventional approach of hybrid modeling. So if you would like to know more detail about this work, please refer to the paper listed here. Thank you.